Okay. Well, welcome everyone. The quorum is present, it is the time, and so this special meeting of the Board of Health is now open. And we only have one agenda item today. I've often looked at the health issues on this island, and there's probably four that I consider really important and significant. One is safe housing. You may wonder why the Board of Health is involved in that, but we often have to deal with too many people living in unsafe conditions, and that's certainly an issue on this island. Opiate addiction is certainly a health issue on this island. I don't have to tell you about that. And clearly, managing and keeping our harbor and our ponds and our rivers environmentally safe actually falls under the Board of Health by statute. And I think the other major health issue, I don't have to tell you, because if you look at some of the statistics, probably 40% of you in this room at some point in your life have had tick-borne disease living on this island. And we kind of struggle with ways to manage that. Well, today we have a potential other approach. Dr. Kevin Asvelt from MIT has a proposal, kind of a long-range idea he has. We're certainly not going to make a decision today about whether we'll actually do that or not, but we have to start the dialogue and we have to understand. And perhaps this will be an option for us at some point down the road. So the way we'll work this today is he'll give his presentation. We'll let the board interject with questions while he's presenting, and then when he's done, we'll take questions from the audience. If we took questions from everyone during his presentation, it would probably get a bit chaotic. So we'll try to keep it under control. But if anyone has something they want to say, or a question or a concern, you certainly will be welcome to do that. Okay, Kevin.
Well, thank you, Malcolm, for the invitation, and to Roberto as well for arranging everything. And thank you all for coming. So the objective here is to prevent tick-borne disease. And tick-borne disease is, of course, an ecological problem. And so in brief, our proposal is to enact an ecological solution. That is, to break the transmission cycle that keeps ticks in the environment infected with these pathogens. That would then reduce the number of human infections because, of course, if no infected ticks means no infected people. The idea is, rather than attempting to control the ticks or control the deer or control the mice that often infect the ticks, instead we would release resistant mice that we have made resistant to the pathogens so that they cannot readily be infected by the pathogens, to therefore remove the source of the infection, which is largely the native white-footed mouse population. So why now? Why this meeting? In brief, this is something new. We are considering deliberate alteration of the local environment. And we should only ever consider that if it offers clear benefits to citizens. We must have the discussions before we run the experiments, ideally even before you run the laboratory experiments. We need to agree upon safeguards and best practices early on before you run the experiments. This kind of environmental alteration that would be a shared impact really should only be done by nonprofit groups. The people running the alteration, the project in the field, should not be the same people to do the monitoring and assessment to see if it is working and to see if it is safe. And the science must be entirely open and responsive to the community from the very beginning.
Can I just, a couple questions?
Yes.
Why do you say it has to be a nonprofit?
In principle, there is no reason. However, there's been a lot of controversy of late over the releases of sterile male mosquitoes to combat dengue and Zika virus by the company Oxitec. If you listen to the town hall meetings of describing this controversy, what comes out over and over again is that people do not trust them because they are a for-profit. And in this case, for this particular application on Nantucket, It would be expensive, but it is the sort of thing that a philanthropic group would likely be willing to fund. Possibly because the later technologies that could extend this to the mainland are very cheap, for reasons that we can get to later.
But you could argue that, while people may not trust a commercial organization, they would still have to go through all the regulatory approval and be approved by an outside agency. And perhaps a private organization would have the funds, because this is not going to be an inexpensive project.
No, definitely not. And it would rely on outside, likely philanthropic support. And again, it would be on us to obtain that support, not on the community here, to be very clear. And yes, the benefit of the private sector is that it can often raise the capital. Here, I believe this would be a pilot that would be a stepping stone towards technologies that I think should not initially be run by the private sector.
Or perhaps if the initial phase one or so was successful, then maybe a private organization would be interested.
Absolutely.
Because my experience is the private organization would first say, I want to see some data.
Absolutely. And what's more, I have no objection to a nonprofit contracting with the private sector to actually run it.
Right.
I just think that in general, people are less comfortable with the notion of contracting out control of their shared environment to the private sector.
Okay.
That's all.
All right.
If the people feel differently, that's great. I have no objection to that.
Fine.
So again, to emphasize, this is only a concept right now. It is something that we are exceedingly confident would work well in the laboratory, or the steps that can be done in the laboratory, and that's why we are coming to you now, before we run experiments. And it really can only move forwards beyond the laboratory if it's embraced by the community. But we would rather come to you now. Finally, and most importantly, it could supplement and not replace current control efforts. This is years down the road, and the problem is now. So this could alleviate the burden years down the road. This is not an excuse to stop existing control measures in any way. So last December, we first held a workshop in which we involved island citizens, admittedly more from the vineyard than from here, scientists, ethicists, leaders of environmental NGOs, Nature Conservancy and others, and regulators, both federal, FDA, and state fish and wildlife, to talk about this sort of thing before we started any experiments or sought funding for this project idea. And the takeaway was that, first of all, field trials of this sort of thing should be done on islands, that it needs to be run by the island citizens, who must agree on veto points that will stop the project, really at points at which you need community assent before the project moves any further. Which particular approach, because there are several different options that can be pursued, should be chosen for the field trials. Where that field trial should be held, presumably on an uninhabited island, not on an inhabited island at first. Which group, who will perform the monitoring and assessment, and then what should be the requirements for a potential eventual local release on Nantucket. Plus, of course, any other decision deemed important by the community that this participants in this workshop did not think of. Of course, there are regulations governing this, both at the state and federal level. That's why we had the regulators present there. And of course, we would have to obtain all standard regulatory approvals. All the efforts would be guided and monitored by local and national ecologists. We have Sam Telford there, whom many of you know. And the trials, of course, would be sequential. Start with uninhabited islands, and only then move up to smaller inhabited islands before anything on the mainland. To be very clear, we are not proposing any approach that would be anticipated to spread beyond the trial site. That is, no gene drive approaches. And the details on that I'd be happy to explain later. But no gene drive on Nantucket, period. Unless you wanted one, but it's not required. So, the relevant problem. You have a large population of white-footed mice here, and ticks that feed on the mice, and also on the deer. But the infections come not from the deer, but primarily from the mice. That is, the mice infect the ticks when they get bitten with the Lyme bacterium, Borrelia burgdorferi, And then when those infected ticks bite uninfected mice, they pass the spirochete back to the mice. And so you have this continued transmission cycle by which the pathogen remains in the environment. And of course, what you care about is the fraction of ticks that are infected, because you only get infected when bitten by an infected tick, which of course has only become infected because it first bit an infected mouse. So this is how you get Lyme disease. And of course, it's also true for the other tick-borne diseases. Similar cycle holds. The mouse is the main environmental reservoir that passes it to the tick and then on to people. So what would happen if the white-footed mice could no longer become infected? No infected mice, in theory, no infected ticks, no infected people. No is a strong word. This is not the only reservoir in the environment. It's just thought to be the primary one. So what data do we have? Unfortunately, there's not a lot in the field because we haven't really had the opportunity to protect these mice against the pathogens in different ways. This is a vaccination approach that actually used the Lyme vaccine that was on the market for people some years ago and is still on the market for dogs. It works on mice, it works on all mammals. And what they did is they went out in the field, this was on the mainland, and demarcated an area, set out lots of traps, caught the mice. When they caught a mouse, they vaccinated it, tagged it, and let it go. And they did this for a couple of years and found that they managed to vaccinate about 42% of mice to the point where they had some degree of resistance. Not complete resistance, but some degree of resistance. And the fraction of infected ticks went down by a quarter. Of course, this was just a marked out field. There was nothing stopping the mice from moving back and forth and in. So they didn't control at all for unvaccinated mice migrating to the area. They just noted that this certainly happened. So it probably would be a greater reduction were it not for that, had they done this on an island, for example. So this is a pretty rigorous study. I more or less believe this. This is sketchier. So this is an oral vaccine. The idea is you feed the mice bacteria that produce the spirochete protein, and that induces an immune response in the mice that should protect them from being infected. Or it's a little bit more complicated than that, but that's pretty much it. And they found that supposedly you got a reduction of 25% when deployed for 2 years and 76% when it comes to fraction of ticks infected. However, and Malcolm has some very great data on, he looked at their original data and what it looks like before you run appropriate controls, and their vaccine wasn't very good in the first place, so their increase in mouse antibody levels, a measure of how protected the mice are, barely went up at all. And they had real issues with their controls, suggesting that the site was just really variable. That is, the fraction of infected ticks just went up and down a lot naturally, so it's really hard to tell how much of it was really due to this. So I'm not really skeptical of this study. I'd much prefer to rely on this one to say that there is likely to be an effect, but that's all that we have. So what we're thinking of would be a little bit stronger. In fact, it would be a lot stronger. If we could make every mouse produce sufficient antibodies, the immune system molecules that provide a protective response after we've been exposed to a pathogen. If we could make every mouse have enough antibodies that it was resistant from birth, so that it wouldn't need to be exposed to the pathogen, it would be born with protection, then the reservoir should collapse. Because when a tick bites a protected mouse, the mouse would not become infected. And in fact, the antibodies go into the mouse, go from the mouse into the tick, and would actually kill the pathogen in the tick. So in principle, the reservoir should collapse. Now, some ticks could still become infected from the secondary reservoirs. That is, there are other rodents that are bitten by ticks, and some of them can also harbor the spirochetes that cause these diseases. But it's a cycle, remember? The ticks bite the rodents and infect them and get infected in turn. If the major group of rodents that are currently the source of this all becomes non-permissive, just spirochetes go away when they bite them, then all of the other reservoirs should also go down. by quite a lot. Not to zero, but down by quite a lot. So that's the idea. How could we do this? So this is the part where we now have the ability in the laboratory to consider doing something like this, and that was not true even a few years ago. So how do we identify the best antibodies? Well, we have vaccines. In fact, we have 2 different vaccines that our collaborator, Lyndon Hu, who also does a lot of separate work with Sam, has been working with. Yes?
Malcolm invited questions as we go.
Yes.
So just to understand about the reservoirs,
Yes.
the other ones were going down. Could you just go through that one more time for me? Thank you.
Yeah, so as best we understand it, the main reservoir for all of these pathogens, so Borrelia, Anaplasma, Ehrlichia, and Babesia, is the white-footed mouse. So it can also exist in other small rodents, in particular shrews, chipmunks, maybe meadow voles. But the mice are the most efficient at infecting the ticks, and they're most efficient at becoming infected when they're bitten by an infected tick. So you have a larger fraction of the mice have the pathogens, and it doesn't harm the mice, by the way. We're sort of an incidental side effect, a casualty in this long-standing ecological war. But the idea is, if you can imagine a bunch of different balls, and the biggest ball by far is the mice, and then you have a few other smaller ones that are the other rodents. But they're all being kept inflated by the ticks, which are sort of hoses pumping air into them, between them, and they're all mutually connected. If you remove the main ball, then the air keeping the other balls inflated goes away.
So sort of to say it as I follow you, the ticks that are working on the mice no longer can get the infection. So even if they bite a chipmunk, nothing's going to happen. So that reservoir goes down because there's less coming from the mice in the first place, or something like that.
That's right. So right now the reservoir in, say, the chipmunks is at the level it is because a lot of the ticks are infected. And most of the ticks are infected because of the mice. Mm-hmm. So if you take away the mice, then a lot fewer ticks are going to be infected, period. And that means you're going to have fewer infected chipmunks, which in turn means fewer infected ticks, and so the cycle. And so it's a self-reinforcing cycle either direction, whichever way you perturb it.
So, Doctor, along the same lines, do these antibodies also convey permanent immunity to the ticks as well?
Good question. So there's going to be 2 different approaches available. You can either protect the mice from the Lyme disease pathogen in particular, or you can try to protect them against the tick bites, which would then protect against all of the pathogens except possibly some of the tick-borne viruses.
Gotcha.
I have some later slides on that one. So there's a few options in terms of that. Yes?
I don't want to get ahead of your presentation. The lifespan of the average mouse who's out there, white-footed mouse who's out there?
Yep, no, great point. So the average mouse lives in the environment for less than a year, survives. The tick life cycle is actually 2 years long. So most of the delay in any impact would be simply because the ticks take so long to mature and bite 3 times over the course of their development. The mice reproduce much faster, typically once every 2.5 months. So the idea is we have these vaccines. We have 2 different forms of vaccines that our colleague Lyndon has developed. One is just against the Lyme pathogen, and the other is against a tick molecule found in the saliva that the tick uses to suppress the immune response so it can keep on feeding. So the idea is you can either protect the mice against the specific Lyme disease pathogen, or you can try to block tick feeding. If you block tick feeding successfully, the tick can't stay attached as long, and so the odds of transmitting a pathogen goes down a lot. And this can block transmission of essentially all of the pathogens except for the viruses. So vaccinate a bunch of mice, test how resistant they are by exposing them to infected ticks, identify the most resistant mice, then pull out all of the cells in the mice that are producing the antibodies. That is, the mouse's immune system actually evolves antibodies that can recognize the molecules on those pathogens or on those tick molecules and raises an immune response to them. So that's how our immune systems work. We actually have specialized cells that evolve molecules that get better and better and better at recognizing a particular foreign molecule. So what we do when we vaccinate something is we just give them a lot of foreign molecule and we say, here, this is foreign. Evolve a way to recognize this. So then we sequence the particular cells to identify the particular DNA strands that have evolved to produce the best and most protective antibodies from the mice that are the most resistant. And then we can pull out the ones that work best in the laboratory. We can actually find the ones that are the tightest binders to the target molecules. And we can pull out a bunch of them. And what we found in tests against other diseases, that if you deliver a large number of different defenses that have been evolved from different individuals, and you put them all into one mouse, that mouse becomes incredibly resistant to whatever the pathogen is. And in fact, this works. You can protect mice against HIV, against malaria, against the flu, against hepatitis. You can even make them infertile by calling down an immune response against key fertility molecules. So it's an incredibly effective approach that we already know works in mice. That's not exactly the same species of mouse as the white-footed mouse, but it's a small rodent. And we know how to engineer small rodents. And the immune system response is very well conserved between the rodents and even all the way out to us.
How do you transfer the chromosomal material that you isolated into another mouse?
So the way they've done it so far has been pretty much entirely deliver the DNA into the muscle. And the muscle cells then receive the new antibody encoding DNA and begin producing it. And they actually use a viral vector in that case that doesn't integrate it into the chromosomes. So the new DNA just sticks around in the muscle cell.
Are you using electroporation? How do you get it into the cell?
You encode the antibody molecules into sort of the shell of a virus, where you scoop out all the virus DNA, and you put in the antibodies as a viral vector. Those studies. That's not what we would be doing specifically, but that's how they did those studies.
How efficient is that?
On any given cell level, very inefficient. But you inject a lot into the muscle, and you're going to get enough cells that they're going to produce a lot of antibodies. So
it's not in the genetic material which is transferred to offspring.
right. In general, people have not done that because there has not really been reason to do that.
Right.
And we didn't have a very good way of doing that until very recently. Now we do. It's this molecule called CRISPR, which is essentially a molecular scalpel that we can program to very precisely cut any DNA sequence we want and insert whatever new DNA we want. So in this case, we would be cutting the mouse genome and inserting these antibody molecules from other mice. So it would be recombinant mouse. That is, it would not be something that would arise in nature, but it would be 100% mouse DNA. We would not be inserting any foreign genes from any other species. It would all just be defenses evolved by different mice. We'd be giving them all to one mouse and giving it the ability to transmit it to its offspring.
So it's an embryo level or a zygote level?
So the way we do it is we actually inject, apologies to all the men in the audience, we actually inject the gonads of the mice. So the sperm-producing cells would take up the new DNA. They would express CRISPR in those cells. It would cut those chromosomes and insert the antibodies. So you would have, and we would actually do this at multiple sites. So multiple different chromosomes would have the protective antibody cassette.
If the two gentlemen that spoke would just afterwards give your name to the recorder, for the record.
All right. So once we have our protective antibodies, we then insert them into the mouse chromosomes, into the sperm-producing cells. When those mice then mate, many of their offspring will then inherit those changes and be able to produce those antibodies, potentially from birth. It depends on how we can program them to start producing them at different times, but ideally from birth. And then once we have them, and we know that they work, that is, we test them again, test how resistant they are with the ticks, Then the question becomes, do they work in the environment? That is, can we get enough of that protective immunity cassette into the local mouse population as to block all of them from becoming infected with the pathogens? And here would be where your first field trial, presumably on an uninhabited island, and the idea is over successive mouse generations, so every couple of months, you would release a large number of mice into the local environment, and they would mate with the local mice, and the offspring would inherit copies of the protective cassette. And you keep on doing this until you've introduced enough mice so that most mice on average are protected. And if you keep doing it, eventually on average all the mice will have at least one. And that's part of the reason why we would add multiple copies, because if you have only one copy in the mouse, then its offspring have a 50% chance of inheriting. If you have 8 copies in the mouse, well, on average they're going to inherit 4. And then their offspring are going to inherit 2, and their offspring are going to inherit 1. So you get 8 times the bang for the buck if you add extra copies. Yes?
After that field trial 1, what are your go, no-go criteria after that?
That's really not up to us. That would be up to the community of field trial 2, which could potentially be Nantucket or the Vineyard would be the 2 best
Yeah, but shouldn't there
candidates.
be some scientific decision about what those go, no-go criteria are? And you do them ahead of time. You write them down before you do the experiment.
Absolutely.
And you either make it or you don't make it.
Absolutely.
Like,
But on the flip side, it's
what would it be, a number of mice that are altered?
I would say rather than some number of mice that are altered, possibly some relative to the number of mice that you managed to alter.
Yeah. How many?
How low can you get the tick infection rate relative to what it initially was? And do you do this on one island? I mean, we already saw from that second study that sometimes you get really variable results. Are you sure that it was not just random chance? Do you want to do multiple field trials? It gets more expensive, but again, I think that this is one where coming up with the funds for this would actually be possible. Now, why? Why would any philanthropist be willing to do this? Well, I could make a big speech about how, oh, wouldn't they love our community-focused approach? And maybe they would, but probably not enough to support that. It's that if this works here, then we could extend it eventually to the mainland. You only want to do that if you have if you know that it's going to work. And if you want to extend it to the mainland, there's a lot more mice on the mainland than there are on Nantucket or the Vineyard. So you just cannot possibly raise and release enough mice to make a difference on the mainland. You need to use a new approach, and that's where we have something called a gene drive, which I'll talk about later. Yes.
Wait, I'm sorry. We have a question out there? Could you come up to the microphone so we can hear you? Because this is all being recorded and minuted, et cetera.
Or should they wait till the end, which is where you started?
Yeah, we'll do this one, and then we'll wait till the end for the questions.
Okay, my name is Ernie Steinauer. Is there some reason to think that this technique is going to increase the survival of these mice? Is it going to infer some kind of selective advantage so it will spread through the mouse population, or is it going to have some selective disadvantage and just going to get wiped out over time?
Great question. The odds are closer to the latter. So that is, in general, we mess with something that is evolved in its natural habitat to optimize reproduction, we're going to break something. In this case, having it produce extra antibodies, it's producing antibodies against something that doesn't really hurt it. Like, as best we can tell, the mice are not hurt appreciably by ticks or by the Lyme pathogen. So we're just wasting its energy. Now, something as big as a mouse, we're actually pretty appallingly energy inefficient, whereas something that's really, really small has to be very frugal, but it's still going to be a cost. So there are no known examples of something where humans have actually succeeded in creating a fitness advantage for a wild organism in its natural habitat. Obviously, if you make something herbicide resistant and you spray herbicide everywhere, yeah, that's an advantage where you're spraying herbicide everywhere. But in the natural, unaltered habitat, we haven't been able to do that yet. I'm not saying it's impossible. We definitely could. But in general, you tinker with something, you're going to break it.
And are you going to use Nantucket mice as your breeding population or wherever you're going to do this at? or just some generic white-footed mouse?
It's another great question, and that's one that the State Fish and Wildlife cares about very much, and they would not let us do it unless we use the local Nantucket population. So as part of the breeding up enough mice to release, we would need to breed them with local Nantucket mice. And that, frankly, we need to do that anyway, because the ones we start with and would add the antibodies to would be easy to work with because they're an inbred population. And that means they're probably not very fit in the wild. So you'd want to breed them a lot so that most of their genetic background will be the wild type ones. And then that also reduces the chances that you're going to mess with something, because then the mice you're releasing would be genetically a match for the ones that they're going to mate with in the wild population anyway. So what would the timeline be? Again, this is not something anytime soon. We think that this is a pretty conservative estimate for when we should be able to confirm that we have made immune mice. This is, again, genome editing in rodents is something we do all the time. No one has done it in a white-footed mouse yet, but again, this delivering into the sperm-producing cells is a pretty reliable technique, and across multiple species. And CRISPR works in every organism ever tested. So we're very confident of that aspect. Sam is currently having the white-footed mouse genome sequenced, so once we have that, we will know what we are trying to cut and can very precisely put the antibodies wherever we want and nowhere else. But the main time delay would be, it's going to take us 2.5 years to generate mice enough for a small island, just in terms of breeding them from those first initial ones. And that's in large part because the first mice you engineer would have a bunch of different copies of the antibody cassette, but then you're mating them to these wild Nantucket mice that don't. And so you have to continually select for the ones that by sheer random chance happen to have a lot of the alleles, a lot of different copies, and then breed those over and over again. So you have some very lucky male mice, and the rest of them are unlucky. So that's 2.5 years to even start the uninhabited island trial. So you're looking at something on the order of 2020. And then once you have that level of population, it only takes you 1 additional year to generate enough mice for a large island trial. But your uninhabited island trial would probably need to run for at least 2 years, because remember, that's the tick life cycle. So you should really start seeing an impact there. And if this works, you should start seeing a significant impact there. And then you can take that into consideration for evaluation and decide when would you want to begin releasing on Nantucket or the Vineyard. And of course, candidates for your should the project move forward stage would of course be at all of these different steps. You probably don't want to start breeding up tens of thousands of mice before you're reasonably confident you're going to be able to at least do the first field trial, for example. Questions on the timeline?
Why would you want an uninhabited island?
Well, if an inhabited island would volunteer to be the first field trial site, that's entirely up to them. It's largely just that we have very little experience deliberately introducing things into the environment that are expected to replicate and evolve over time. Now again, we tinker with something, we generally break it. Anything that's going to happen, probably it's just going to be that the population is going to be selected to remove this resistance. That's going to take a long time, decades, if not longer, in all likelihood. But if anything, it's only going to go down. But we don't know what else will happen. And that really gets to, I'm going to skip ahead a little bit. It would depend on what you're doing to the mice. If you're just protecting them against the Lyme pathogen, the spirochete, you're probably not going to impact any other species in the environment, because you're just going to be lowering the burden of the spirochete. Most of the things the spirochete infects, it doesn't really hurt, as best we know. It seems to hit us and our dogs because we're new here by evolutionary time. Most of the things that it does infect have sort of evolved an uneasy coexistence. So that's pretty ecologically safe. But if you immunize the mice against the ticks, then your tick population is going to go down. That might have impacts on other species that feed on the ticks, that get preyed upon by the ticks, etc. Now, right now, to be fair, the tick population is much, much higher than it would be naturally, because there are more deer and there are more mice. So also, there is the question, does the community welcome fewer ticks on the island. It would be closer to the natural level. But it might have ecological side effects that you might not anticipate. So in general, you want to run that sort of thing on an uninhabited island first. That said, that's the sort of thing that's entirely in the community's hands, not in mine.
Okay, go ahead.
And then, of course, option 3, which is probably the safest from a long-term perspective, is just do both. Make sure you hit the Lyme pathogen, the Lyme spirochete, but then also target the ticks as well to inhibit transmission of all of the others. Because in this case, it's not an either-or. We can really have both. For funding? Yes.
Go ahead.
One more question. When you said respect to your comment about more deer, you were referring to Nantucket when you said that?
In general.
what?
So nothing we're proposing here would touch the deer. There's a long-standing, I understand it, this is not my field, dispute over the appropriate level of deer in the habitat. But my understanding is that right now in many areas part of the reason why there are so many ticks is because of the deer.
Because there are more deer than someplace else, I guess is what you're saying.
Well, there's more.
I'm just trying to, because we have Lyme disease in Minnesota and Washington. Do they all have more deer as well? So I'm just trying to understand the use of the term. And does it apply just here or any place that there's Lyme disease?
The deer population seems to correlate with the incidence of Lyme disease.
I understand. It seems to.
I'm trying to be, seems to.
Okay, thank you.
And it's admittedly complicated because it's also, again, the deer are not hosts for the spirochete. So it's also a matter of are there the mice or there are some other host,
Right.
reservoir host. And so here it's the mice, so it's a combination in all likelihood. But again, Malcolm is far more of an expert on this than I
I don't know
am.
about that, but
Really defer to Sam.
there's an estimate made in 1900 there were 1,000 deer in Massachusetts. All right, we have 2,500 right here ourselves, so it's not probably natural to have this many deer, like you're saying. If we didn't have all the deer, all the mice, we'd still have Lyme disease, but not at these
Right,
levels.
and my only point is it may not be just here. I mean,
Oh no, it's not
there's
just
more deer lots of places.
Yes,
Yeah,
Yes.
okay.
the whole eastern United States has more deer.
And it's probably some combination of we rejigger the landscape to favor the mice because they prefer fragmented woodland habitat, and so you get many more white-footed mice relative to all the other small mammals that are not as good a hosts. And then you get more deer, so you get more ticks, and it's the combination of these, and it's been getting worse and worse.
And the deer serve as the host, right, the blood host for the tick. It is
Right.
part of the cycle.
Right.
Well, they
Yeah.
we're talking about mice today.
Yeah.
I do
Yeah, we're
not
focusing
want
on
to
the
start
mice.
Deer.
Not dealing with the deer.
If we start about deer, we'll be here all night. Let's just talk about mice.
So initially we were relying, so my research group at MIT just launched recently. And so we have substantial support from MIT for our initial laboratory work, so we can begin experiments in developing these mice based on that. And we also have several potential sources of philanthropic funding that we're looking into. But we have not yet secured funding for this project very deliberately, because this is a case where we believe it's appropriate to go to the community before you have the funding or run the experiments. Scaling up, definitely going to need philanthropic funding at a fairly large scale, typically looking at the low tens of millions of dollars for projects of this scale from the multiple stages of field trials. And the reason is the sheer scale. So there are somewhere in the probably low hundreds of thousands of white-footed mice on Nantucket. So you want to get a protective antibody DNA cassette into the vast majority of those. With conventional methods, you have to release a comparable number of mice over a period of time. So you don't want to release too many because then you overburden the environment and more likely to cause ecological disruption. And how many mice do you really want to find in your house? So you could release immunized mice constituting maybe 10% of what you think is the local population. You do that every couple of months, so every mouse generation. And that's not too much of a burden because the mouse populations normally fluctuate by at least 50% over the course of a year. Sam, please interrupt me to if I ever say anything incorrect on this, but with this approach, the mouse population would be at most 150% of normal, and that's assuming that they could all survive. And of course, there are natural limits on the population, so you release extra ones, it's probably just going to die back down to the appropriate level, what the environment can support. And you can absolutely use bait stations around actual commercial and residential areas because the bait stations are not going to discriminate between the introduced mice and the wild mice. What you care about is the total number of mice. So you can still use control, and that should actually be part of the project. So questions for all of you. Is this something that you might at least want to think about considering? And what criteria would need to be met for you to consider releasing it? Should the mice be immunized against Lyme, ticks, or both? And again, this is the sort of thing where we would start making them in the next year or so. And we could try both, but better to know in advance what you would prefer. Where should those first field trials be held? Which islands? I mean, if they're uninhabited, then who's going to pick? I mean, it should be obviously in consultation with ecologists like Sam, and the regulators have to approve, but really you should have the choice about what you would be confident in. And who's going to do the monitoring assessment? Because you might have heard of some reproducibility crises in certain fields of science, particularly psychology. And a lot of that comes from the fact that really the last person you want to test a hypothesis is the person who came up with it. That's the single most biased person there is. Why do we have them do it? Science is broken in that sense.
Well,
so
in my field, in human experiments, we now extensively use a data safety monitoring committee, which is a group of independent people not involved with the investigator or the sponsor or essentially review it.
Exactly.
You need something like that.
So you need something like that, and ideally you should come up with independent funding. Maybe government funding to ensure that there's no bias because, what do they have? But ideally independent group, independent funding, maybe multiple groups, could be local groups, could be people you bring in, could be private, could be nonprofit. Now, there are a handful of journalists here. It's fair to say that there will be outside interest and media attention should this go forward on Nantucket due to partly the community-driven approach. It's not entirely usual to really go to the community first and try to have a grassroots community-owned project, but also because there is the possibility that if it worked, we could then have discussions about spreading this resistance to the mainland populations. Not using this approach, but by using the aforementioned gene drive to spread the trait through the population, which is the one way that we do have to potentially cause something to spread in the wild. Otherwise, in general, we can't do it. So to show you what might be the complete cycle if this worked, you get to here. If you want to scale up further, even to something like a town or city on the mainland, because you would have the constant invasion and pressure from the surrounding mouse population, diluting out the effect of your antibodies, and the sheer cost of releasing enough mice to make a dent in a mainland population, that's just not really feasible. But we've invented this thing called the daisy drive, which is a way of causing in this case, the antibody cassette, to spread from one initial mouse release to a few hundred, and then it stops. And so that could let you do local communities on the mainland affordably alter their local population and immunize them while releasing many fewer mice. And then if that worked well and people were happy with it, then you could eventually consider, after you've talked with Canada and Mexico, building what is called a full gene drive cassette, which would spread it through the entire white-footed mouse population and reduce Lyme disease throughout the Northeast. That's why there would potentially be outside media interest in this project, because of the very long-term downstream applications that would potentially ensue. Not saying that they would, but if it worked here, then those questions would begin to come up. And that's partly why we are so adamant that we do this differently from the very beginning in terms of being community-owned and community-driven. But again, this would not affect Nantucket in any way. There is no need for any kind of drive system on Nantucket because the size of the island is small enough that we could just produce enough mice and release them that you don't need one. And if you wanted to, a daisy drive system would let you spread the resistance faster with fewer mice. However, the drive systems do require foreign DNA, which some people are uncomfortable with. So on Nantucket, we wouldn't need any foreign DNA at all. It would just be 100% native mouse DNA. If you wanted to spread it on the mainland, you do need some foreign DNA. And finally, of course, this kind of local and grassroots approach made by decisions using New England town hall democracy could be a model for projects elsewhere in the world tackling other diseases that could help communities decide questions of those. So here's sort of the stopping point. I have more slides to explain gene drive if you're interested, but again, that's not strictly relevant to this project.
If you have questions, I have some more. What I'm going to do is we'll open it up to the audience, and if you want to ask a question or comment, if you just come forward, identify yourself to one of the microphones. Sam.
Sam Telford, Tufts University, someone who's been working here for 30 years on the problem of reducing risk. The reason I'm here with Kevin, Kevin approached me in November about this idea. And you, as a community, have rejected our suggestion for many, many years that you remove a large portion of your deer population. I still insist that if you were to have the guts to do it, you would have no Lyme disease here within 20 years. But that's not going to happen because of all the socio-political forces that we know operate on this island. So the reason this approach has interested me is that besides deer reduction, we have no other what I call long-term public health interventions in the pipeline. I distinguish public health interventions into long-term or short-term. Short-term are ones where you have to do it periodically on a daily, weekly, monthly, or yearly basis and forever expend resources and energy. Things such as buying a can of repellent every few weeks, things such as tucking your cuffs into your trousers every day. Those are short-term approaches. Insecticide treatment stations. Short-term approaches. You will have to do them forevermore. You stop doing them, the problem comes back or still continues to plague you. Deer reduction would permanently make this place a better place for your children's children. But that's not going to happen. So we need something else in the pipeline as a long-term solution. This technology, CRISPR, is fantastic. Kevin is going to make these mice. And so I think that this is a possible tool to complement our short-term intervention methods, assuming that there's going to be no focus on deer. And even if there were, the two would go hand in hand because most people don't understand you need to produce ticks. That's deer producing the female, letting the female take blood, and each female laying 2,000 eggs, and then you need to infect the ticks that result from that, the baby ticks, and that comes from things such as white-footed mice and birds and so forth. There are two processes, chicken or the egg, and if you can attack both, deer reduction and modify mice, that's even better. So that's why I'm here, and that's why I'm going to be very happy to support Kevin in the ecological aspects of this project.
Okay, thank you very much. Dr. Lepre, I'm sure you have a question or two.
Just one.
Well, I will make a statement, and having dealt with the same problems that Sam has mentioned, having been just about ridden out in a rail when we had the extra season in February, deer reduction became a moot point after Bambi came out, and that was in 1942, okay? Unfortunately, the white-footed mouse is not quite as cute, and I think this approach really makes sense to me. I think the state wanted to do more deer reduction, but quite frankly, I think if we were in a parallel universe and able to get the deer herd down to 15 per square mile, I think that you would really reduce the tick-borne illnesses. The problem is that if you get them down to 15 per square mile, nobody's going to come out hunting, and all of a sudden we're going to have 2,500 deer 2,500 deer on the island. So I think this approach makes an awful lot of sense to me, and I'm really excited and enthusiastic about this. I see an awful lot of patients that have had Lyme disease and the variety of the other tick-borne diseases, and I think it's incumbent on us to do something. As Sam had mentioned, most of the approaches we've had have been short-term and have to be repeated every year because all of the does are having twins and some of them are having triplets. It's an ideal place to be a deer. If you look both ways and you don't get it from a Range Rover and you go on the posted land when the first shotgun goes off, you're all set. If we have the 4 posters, it's going to be a ton of corn a year and all of those problems. Those are recurrent problems.
Right.
This seems to address the issue in a very reasonable and very interesting way.
Thank you. Anybody else? Questions, comments? Roberto?
I have 2 questions. One of them is, how are you going to socialize some of these mice to prevent them from being eaten by the harriers and the owls and everything while they're here? And then secondly, evolutionarily aside, I'm assuming you're using a promoter and a terminator inside the gonads of these mice. Are there any chances of that promoter becoming wild type outside of the mice once they start procreating in other animals other than just the mice?
So the first question is a good one, and there we're going to be largely relying on Sam's expertise in terms of how can we rear mice, white-footed mice, at large scale. That's admittedly, I'm primarily an evolutionary and molecular biologist. I specialize in how do you do the molecular scale editing and how is it going to evolve and change once you do put it in the wild, which is the really important part. I'm not an expert in mouse rearing in particular. I have colleagues that are much more expert than I, and I would have to leave it to them. But
there are
ways of breeding large numbers of mice. In terms of how you would do it practically, especially given it's possible that there would be some fairly stringent regulations on where you could do this. I don't know, maybe you would. Right now, international shipping is basically plummeted due to the commodities crisis. It'd be really cheap to just rent a tanker and breed a ton of mice in the middle of the ocean. And as for socializing them to be, a large amount of that actually does seem to be genetic.
Okay.
So have the school kids set out traps and capture live mice and send them to us, and we'll interbreed them. We'll interbreed with them so their offspring become immune, and then we can release them. And great community project. So the other one, so molecular biology side,
yes,
we need to express the antibodies. We're not going to do it we're not going to use it using the normal signals that the immune system does because it has this amplification effect. Like it needs to identify the ones that are the best and start producing lots and lots and lots of those. And we don't really want that. We already know we're starting with the ones that work really well. We just need to make them. So we would use a different signals to tell the mouse, produce these. And we would just use signals taken from the mouse genes already. So we would use native mouse promoters and enhancers and 3' untranslated regions to ensure the appropriate level of expression. And we would just choose ones that would work for a given cell type. I'm consulting with some of the people who have done a lot of the work in the European mice as to what is the best cell type, and then we would need to test the different levels and see if make sure that we're not harming the mice in terms of their ability to compete. It'd be a matter of we were going to make a bunch of versions and see which one works best, combination of resistance and cost, and go with that. But they would all be essentially native DNA sequences taken from the mice.
Okay. And sorry, one follow-up question. The antibodies that you're looking into promoting into creating into these animals, do they protect against any other Borrelia species or Ehrlichiosis or anything like that?
So the antibodies against the Lyme spirochete would be against a particular molecule that the spirochete expresses when it's in the tick.
Okay.
And that's the one that the former now pulled Lyme vaccine, the one that you can still get for your dogs, works off of. And it does vary between the species of Borrelia, so it wouldn't protect you necessarily against Miyamotoi.
Okay.
We could use that one and generate antibodies against that. That's one of the nice things about this approach is you can always add more. There's no real cost to adding more. But we would probably not be protecting against the Miyamotoi. We would just do the ospe for Burgdorferi, the Lyme pathogen, mainland pathogen. But targeting the tick salivary proteins, because it disrupts feeding, disrupts transmission of all the pathogens. And from an evolutionary perspective, I'm more comfortable with that one. Because whatever we do, even if we really think we're clever and copy a natural system like a gene drive to spread something, in the long run, natural selection always wins. And the bacteria, the spirochetes, reproduce a heck of a lot faster than the ticks. Remember, the ticks reproduce every 2 years. So the odds of the ticks evolving around a defense that we put in the mice against ticks, that's going to take a really, really long time. Whereas if you just protect against the spirochete, then you're going to select for resistance. I don't know how fast. It's going to be probably not sooner than a decade, but a decade after all of this amount of effort, you really want it to last longer than that if at all possible.
So it is possible that we might experience a bottleneck event, and then whatever comes out of the other side of the bottleneck will be stronger?
Oh, in the long run, yes.
Okay.
It's just as long as that happens more than 100 years from now.
Well, even this project, most of us won't be here for that.
Exactly.
Well,
You'll have a whole new board, hopefully.
And if the sea levels keep rising, the island might not be here.
I know. So, actually, that's good. Any other comments? Yes, please.
My name's Larry LeCain. I live out in Nantucket. We got our share of ticks out there. The first question is it possible to patent when you come up with the super formula for the mouse that's not going to carry any disease? And can Nantucket benefit from that?
it's an interesting one. I believe, however, that the answer is no, because the Supreme Court has ruled that you cannot patent anything that is a pure product of nature. And in this case, what we would be isolating would be a pure product of nature. Now, you could argue that the combination of the particular antibodies, even though they're all pure products of nature, is unique, and that the combination of all those different products of nature to express them all in a particular cell type and resulting in a newborn mouse that is resistant is not something that is product of nature, but you would probably have to take that to the Supreme Court.
I thought they ruled already for Dr. Chakrabarty to patent a life form.
Yeah, they ruled against Myriad in terms of essentially yes.
But they ruled in favor of the ability of a person that comes up with a way to do it, that creates a new life form, to hold a patent on that.
But they've also ruled that you cannot simply take something that already exists in nature.
Oh, so you can't modify it. You have to come up with a whole new thing.
You have to tweak it somehow so that it's different from what is natural. And in this case, would be using, again, the point would be make mice that are 100% native mouse.
So we're just
different combination.
Selecting a trait that's preferred.
Yeah. Now you could argue, again, we're shuffling it in a way that might be unique, but all the pieces were there, so it would be a tough argument to necessarily get it covered.
Because assuming success, and I'm hoping for success, everybody's going to want this. And I bet Lyme, Connecticut would be the first place to go. Let's do it here. To get their reputation back.
Yes. So I think you're absolutely right about that. Now, the complicated bit is that it depends on which. So remember, the mainland would have to do a cheaper approach. They just can't afford to raise and release enough mice. So they would need to use one of the drive systems.
Yeah.
And the Daisy drive version, you might be able to extract some commercial revenue with that. Again, I would prefer that all of this stay nonprofit for a while. But it's certainly true that the full drive version costs, well, very little because it spreads itself indefinitely.
Okay.
So it's really hard to extract commercial revenue from that. So I don't understand enough of the legal situation. I think it's unclear as to whether or not you would be able to patent that. As a general rule, universities nowadays patent everything they possibly can. And in general, I'm okay with this, even though I tend to favor, as you can imagine, nonprofit approaches, because you really have to own it in order to give it away. And if you don't patent something, you always run the risk that even if it should be in the public domain, someone with a really good lawyer could patent it anyway and yank it out from under you. And then you can't use it for the public good.
That's why I say this. I think we need a lawyer.
Well, that's the exact reason you need to patent this, whether it's a valid patent or not. Everything changes in time.
Yes, absolutely.
Yes.
So in essence, this is a recombinant wild-type mouse.
Yeah, the technical term is cisgenic. That is,
Yeah.
all the genes are from the same organism. You're not moving in anything from any other organism. You're just moving them around a bit.
All right, that makes sense.
Okay. Any other comments from the audience or from the board? Sure, Dr. Tim.
What was the foreign DNA you would insert in the drive, not the daisy drive, but the other drive? I mean, why would that be particularly objectionable since nobody's eating these mice? At least nobody too fat.
Well, it's the same for both of them, and it's the same CRISPR system that we use to put DNA in. It's just here's what we would do for the native mice. This would be your antibody cassette. You just use the CRISPR system to cut the target site where you want it to insert it, and then the cell just uses your introduced DNA as the repair template, so it perfectly copies it in. And CRISPR, we now have variants that will only cut the exact sequence you want. They will not cut anything that matches, that has even one base mismatch. It has to be a perfect match to the sequence. It's incredible, and it always works. The way you build a drive system is, if that's your pencil that lets you erase the original gene and rewrite in a new version, if you encode the ability to make the pencil along with the change you want to spread, in this case this would be the antibodies, this is the CRISPR system and the instructions telling it where to cut, you put this into the genome, this is your full CRISPR-based gene drive system, because now your mouse produces your antibodies. And of course, once one copy is in, it will cut the other copy, because you have 2 copies of most genes. And then the magic happens when it mates with a wild-type mouse, because 2 copies means all of the offspring are guaranteed to inherit a copy. And then, of course, it has the ability to do genome editing. So it will erase the original version and rewrite in your antibody-protected version. And then all the offspring will inherit again, which means genome editing happens again in the next generation, which means every generation you erase the original version and write in the version that has the protective antibodies. And it keeps doing that until you've protected every mouse in the population. Again, natural selection is eventually going to erase those antibodies, but it's going to take a very, very long time in this case. Thousands, tens of thousands of years probably, because you would have spread it to almost all of them. But this is, frankly, a problem. How could you even do this? First of all, do you even have the right to build one of these things in a laboratory, given that if you screw up and it gets outside, it's going to do this and affect the environment? I say no, of course we don't have the right to do this. Certainly not without telling people. And the problem right now that we're fighting against is, right now there is nothing stopping me or anyone else from just building one of these things. Like, it's illegal to release it, but if it escapes through a hole in the wall, and you can do this in flies and so on. So right now we're trying to ensure that nobody can just do this, and that everything anyone needs to do with this needs to be done in the open in the light of day. So that's my current crusade. And it works now in yeast and flies and several species of mosquitoes. But, okay, even you deal with that, you only do everything in the public, there's backing for it, say against malaria, something that's really an clearly awful problem and you should do something about. How do you test it in the field to make sure that it works? Like, what we're doing here, you're releasing the mice that have the antibodies, and there's no drive system, so it's not going to spread. And you do it on the uninhabited islands, you figure out, is there anything going to go wrong in the environment? With this, you put one organism out there with this, and there's a greater than 10% chance that it's going to start spreading. So this intermediate version that we came up with, I mean, the problem is you can't run these field trials, and what if different communities disagree? So we came up with this version called the DAISY drive. Actually, the paper just came out today in preprint. But instead of having all the components in one piece, so it can spread indefinitely, it has everything it needs, you split them up. So they're in this daisy chain where the C1 helps the B1 to replicate, and B helps A to replicate. So in this case, A would have your antibodies, and the others would just have components of the CRISPR system. And that means that there's nothing helping C. So C is neutral, and in fact it's going to be weeded out slowly. So it stays there, but when it's around it helps B, so B increases, and then B helps A, so A increases fastest of all. So you can sort of think of it as like a genetic booster rocket, in that each of the lower components help the genetic payload, the antibodies, to spread through the population. Over time, but it's fundamentally temporary and limited in terms of how far it can spread. So you can tune the effect by adding more booster stages to the rocket, more links to the daisy chain, so it will spread through more generations. So this is your just releasing a single mouse with an antibody. This is the comparative power of releasing a 5-element daisy drive driving the same thing. So the only foreign elements in here are the components of the CRISPR system. But to a lot of people, that matters. Like the notion that you're introducing DNA from some organism other than the mouse really does matter. So from a scientific standpoint, you run the tests on an island. Does it have an effect? Yes or no? It's possible that, a hawk that eats this mouse, maybe there's some sort of immune reaction to the CRISPR system. It's the sort of thing you would need to test to find out. But that's why you never test the first drive system. So again, it's this latter stuff, these latter stages, that we are so adamant that this has to be done differently. This has to be community-driven from the outset, because the consequences of going forward the old closed-door way are not viable anymore once we have this technology.
Okay, thank you. Now, so essentially you can start your lab work now, and you're planning to do that?
As a young scientist with a fixed startup package, I'm reluctant to do it unless some community says, we might at least think about this.
Well, I think we'd think about it.
Okay, well then we are likely to move forward and do it.
Isn't thinking easy? I mean, it's like free.
I mean, is this board, is there...
Of course we'll think about it.
Is there any of the members here of the board would have any objection? I mean, because to me this is... I think this is quite exciting, but what I like about your approach is you're taking a very slow step, safety ways with oversight. So I can see the next major decision that we would have to make on this island is once you're done, I assume you're going to take some of our mice home with you?
Eventually, yeah. They get upset when we take wild mice in because they often have pathogens and the mice ones we engineer. But yeah.
At some
eventually.
Point, and
for sure,
the key decision point is it's like any project with go no goes. That are you successful in the lab? The next step is the study on the island, and then we would have to know what the clear go no criteria were of that first field trial to make a clear decision.
Yes.
In my business, we've always done that. So you're not there when it's done arguing about it. You say you have to do this, 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 and that, and that.
Yep. Set a set of criteria that have to be met.
I think we, as a board and scientists, and we have Dr. Weld and we have Dr. Lepre and all kinds of great minds. At some point down the road, would clearly have to establish those and be accepted by this board and the island. So I don't hear anyone yelling and screaming, don't go to your lab with this. I think that's the right thing to do. But clearly, before we would support going to the first field trial, we'd have to see the results. And then after the first field trial, that would be, before it would come to the island, we'd have to have those criteria. So, and you have funding for your lab work?
For the initial steps, yes.
Yeah, and the CDC or the state, I think, would be great sources of funding down the road.
Yeah, actually, the white-footed mouse is a new, exciting model of aging research because they live much longer than the European mice. So they probably want us to develop genome editing in any way for that reason.
And then in your oversight, you really need, how do you see this oversight group? Or I'm still a little fuzzy on your project management of this project.
Well, so am I. Honestly, I was going to kick it over to you.
You're
in charge. What kind of oversight do you want? I mean, this is really up to you, not us.
I have some ideas that I have to think about, and I can talk to you about it. I mean, in my other life, I did develop things over time, so I have a little experience in that. So, what I would suggest is I hear no objection for you. I think we'd be very interested in this project as long as it met safety criteria as we went along. And I don't hear anyone objecting to that anywhere. So I will think about some project management ideas and talk to you.
Sounds
Okay,
great.
on that. Any comments? Anybody different opinions here?
I don't have a comment, but I have one more question on the slides that you just brought up.
Yeah, please.
Sorry.
My name is Danica Connors. I'm an herbalist on island, and I help people with Lyme here. And I came here thinking I would say absolutely not. I'm the first person to go, no way. You tinker with the environment, you tinker with Mother Nature, you tinker with any of those possibilities, we're going to break it, we're going to screw it up, there's going to be a trickle-down theory, hawks will be affected, we'll be affected. What are you going to do? Change the structure of a spider, a mosquito, eventually all of these things that pathogenically pass all of this stuff. And, you know, we're seeing signs of things being passed in vitro. This is much, much, much larger than the white-footed mouse. This is such a tiny, 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 tiny spot. But with all of these no-goes that you're talking about, with the level of how careful you're coming across and wanting to be, and I love the fact that you're a young scientist putting your foot down and going, I want this to be a non-corporate thing. I want you guys to give us all of these safety structures before we even move forward. Actually made me go, you know what? I want to see where you go with this. Even I said, I want to see where you go with this. So I'm actually really excited about this little spot that I thought not in a million years. So thanks for this attempt.
Well, thank you for that. I think we definitely need a new approach.
Yeah.
That's good.
Roberto, you want to finish?
Just one last question. We're altering the phenotypic expression of these mice by inserting a gene into one of the two autosomal pairs. By creating that, aren't we creating a knockout gene at the same time? And is that knockout also not coupled with a potential autosomal abnormality that will proliferate in the environment?
So we would choose the insertion site specifically because, as best we can tell based on what we know of the lab mouse, the lab rat, and humans and other organisms that we understand their genetics well, don't do anything. Okay. I mean, most of our genome doesn't apparently do anything. I mean, it does something because it soaks up transcription factors and binding proteins so that everything is the appropriate level. But so we would just pick neutral sites that don't matter. There's no reason to pick anything that, if there's even the slightest chance that we think it might be doing something, just don't hit that site. We have the whole genome to work with.
Okay. And it's nothing with the exonuclease that new splicing sites that might be created through this, or are you already looking into all that?
So in this case, I think we just stay away from anything that looks like it might produce a protein in any way, shape, or form, just to make sure. Yes, there's bizarre trans-splicing. Biology is complicated. We are still learning all of this. This is why we need to be very, very careful. But in this case, if you make the mouse and you don't see an effect, then you don't see an effect. But we can also test for those sorts of things. Is this, we can pull out that particular antibody because we know the sequence of it. Is it interrupting? Is it splicing with some other transcript? Is it interfering with some other gene? We can look for those things.
Okay.
Excellent. And you just have to do it.
There's a comment from, we'll go David first, then Sam, and then we'll close.
Thanks, Malcolm. So thank you for this presentation. I'm really excited about the openness that you talked about.
Identify yourself, please.
What's that?
I know who you are.
Hi. Oh, David Gagnon, Executive Director of Mariah Mitchell. So my question is, you did mention that you already have some funding, and I'm just curious to be really open about it, exactly what that funding is that you have at this point to get started. So thank you.
So I'm lucky enough to be a faculty member at the Media Lab, which is a place for people who are antidisciplinary and like to challenge the status quo way of doing things. And in general, when you start a new laboratory, you don't have any federal grants or philanthropic support or anything, so your university provides you with a pile of startup funding to support students. So for example, Joanna here is a graduate student who would be leading this project, and so she's supported by the Media Lab because they provide student support for the first several years. And so it would be that startup funding that would be used. It's because we are a new laboratory, a new research group, that we have that sort of just lump sum funding, spend it on whatever you want from MIT that lets us begin the project like this. And then we would probably also apply for some federal grants, look for some philanthropic donors that might be interested due to the long-term potential and really forging a new and open and responsive science.
Yes, both of you.
Oh, actually, I should add one more thing. The Media Lab does have an unusual funding model. I'm afraid that a lot of the funding is from corporate sponsors, but it's into a general pool of corporate sponsor funding. It's divided up among all the Media Lab faculty. Most of them do stuff that's corporate relevant. Our stuff, not so much, but they tolerate us anyway.
I can't resist taking the opportunity to emphasize the last line there. We are right to be very optimistic about Kevin and having maybe 20 years from now something that will have reduced transmission on this island. But that doesn't mean that we can give up what we know today should be done each and every day. Check yourself for ticks, wear repellents. If you're sick during the summer, go see a physician. And to continue to think about things that you can do to clean up around your yards, around your schools, clean up the environment, support brush reduction, and think again about deer reduction, because mouse manipulation and deer reduction can go hand in hand. But I remember when the Lyme disease vaccine trials started, I ran them out here back in the mid-1990s. Everybody was so enthusiastic about a human Lyme disease vaccine being on the horizon that everything else just went in the trash can. Nobody bothered to check themselves for ticks. Everybody stopped talking about deer reduction, and it was a setback for public health in general. This is the pipeline. Malcolm can tell you how much optimism there is for a drug pipeline and what might be in the pathway to approval. And so we need to emphasize that last line there in the interim. But we're all excited about the possibility of what might happen 20 years from now.
Yes, thank you. And it's a great idea, but there's no guarantee this is going to work, and the probability of success at this point is probably actually low. All right? So we had one more comment.
I'm Jim Lentowski, the Executive Director of the Nantucket Conservation Foundation. We own about 30% of the island. So this is an exciting idea. As soon as it's out being discussed in public, people are going to say, this is some secret science that's going on. And I think this really does beg for the need to have a dumbed-down version of what has just been discussed this afternoon available to the public, so that they start understanding what you're talking about before they start having suspicions about this being Dr. Strangelove in the laboratory and we're going to be dropping mice out of parachutes into the island. Thank you.
Thank you. We'll work on that. And I just want to emphasize to everyone, this is just an idea and a concept, and we're a long way from parachuting mice in Nantucket, okay? A long way. We have hurdles to go through and decisions to make, and if we come to the point where we're going to start parachuting mice, there'll be a lot more discussion. Okay? We just want to get to that point we can make the decision. Anyone else? Well, Kevin, thank you. I think this is really exciting, and I appreciate your comments on deer, but I don't want to get into that today.
I almost wanted to close with that.
Go ahead. Any comments?
Well, no, I do find it interesting that Dr. Telford's comment was the island has rejected deer reduction, and therefore a possible solution, it seems to me the corollary, we've accepted Lyme disease for a lot more people than we need to. So that seems to be the corollary that I get out of that. Different from the subject of the presentation.
Thank you. You're here. All right, hearing no objections, and we will adjourn this meeting.